Good afternoon and welcome to today's webinar titled Introduction to AQUATOX. This webinar is sponsored by EPA's Water Quality Modeling Workgroup. I am Brenda Rashley with the U.S. Environmental Protection Agency and I will be moderating today's webinar. Thank you all for joining today. First, I'm going to briefly summarize some of the features of today's webinar. We hope you will have lots of questions. Please submit your questions in the presenter questions box in the control panel. If you have any problems viewing the broadcast, just let us know in the Adobe Connect help box, and we will post an answer in that same box. The webinar will be archived for later viewing on EPA's Water Quality Modeling training page, and we provide that archive link for you at the bottom right of your screen. At the end of today's webinar, you will be able to obtain a certificate of attendance for participating, and we'll provide directions for downloading it at that time. The materials in the webinar have been reviewed by U.S. EPA staff for technical accuracy. However, the views of the speakers are their own and do not necessarily reflect those of the U.S. EPA. Mention of commercial enterprises, products, or publications does not mean that U.S. EPA endorses them. The purpose of the webinar series is to help water quality professionals better understand water quality modeling and how models can be used to solve problems facing water quality regulators. The target audience for this series is a water quality professional who may use or want to use or review models as part of their job, but who do not necessarily have a modeling background. As such, we are assuming that the audience has a basic understanding of water quality science and concepts, but has limited direct modeling experience. Today's webinar is the 21st in our water quality modeling series. All of the past webinars have been recorded, archived, and posted at EPA's YouTube channel. Links to the archived files are available on our modeling workgroup website. The URL shown at the bottom right of the screen. Further, in a few weeks, a copy of today's webinar archive will be posted on the same website. We encourage you to listen to any of the previous webinars or share access to these files with your coworkers. I'm very pleased to be able to introduce our speaker today, Jonathan Clow. Jonathan is founder and manager of Warren Pinnacle Consulting, Incorporated. He has served as a consultant to EPA since 1994 with special emphasis on developing object-oriented environmental computer models. Since 1995, he has been the primary programmer for EPA's AQUATOX model. I'd like to hand the floor over to Jonathan to kick off today's webinar.
Thank you, Brenda, and good afternoon, everyone, or depending on where you are in the country, I suppose. Good morning. So, I'm going to talk about the AQUATOX model, and it is a big subject because it has been under development for many years. It has a very wide breadth as far as these models go, but one of the things that I'm going to display is how we're in the process of breaking it into smaller pieces as well, so that to increase interoperability with other modeling systems and the like. But the basic, in a sentence, definition of what AQUATOX is that it is a model that represents the combined environmental fate and effects of conventional pollutants, and that includes nutrients, inorganic sediments, and toxic organic chemicals in aquatic ecosystems. And so I think the multi-stressor piece is one of the key pieces to take home with regards to this question. So we can't really talk about the AQUATOX model without talking about Dr. Richard A. Park, or Dick Park as he's known to those who have known him for many years, now retired. But he has been an ecological modeler and started the firm Eco Modeling, which was a good reflection of what he did. but he's been an ecological modeler and an academic and a consultant since the early 1970s working on these models. And he, in the early 1980s, realized that there were a number of models that were being constructed simultaneously but side by side. And he, so what his idea, his brainchild, was to take existing developing and improving eutrophication models, modeling nutrient impacts and changes, combining that with a food web bioaccumulation model that calculates tox trace, for example, which calculates the uptake of organic chemicals into the food web, and then also adding ecotoxicological constructs. You could also look at the effects of nutrients and organic chemicals at the same time. So the model has been in development for quite some time. There have been multiple releases from 1988 through the current date, and it's still under development. And release 3.2 is the latest version that EPA put out in 2017. So at its heart is a bioaccumulation model, and here's an example of a conceptual bioaccumulation model for a river. All these components we can find within AQUATOX represented as a state variable, which means that sort of the biomass or the concentration of these components within can be calculated over time. And so it includes organic matter in the anyway, it includes organic matter in the suspended in the water, then the invertebrates, the lower level infaunal and epifaunal invertebrates, water column invertebrates, forage fish, and also game fish. And what's not shown here are nutrients. And for a pond, you have a very similar set of state variables, but everything changes potentially depending on the salinity and also depending on where you're located. So the thing about an ecological model is you have to represent the ecology of your site, and so you can have very significant changes based on whether you're modeling still water or whether you're modeling faster water or whether you're modeling salinity. So there's a lot of flexibility. There are databases of parameters to model dozens and dozens of animals, plants, and chemicals built in. So back in 2004, I worked with John Imhoff and a team with Aquaterra Consultants to kind of understand, to look at different bioaccumulation models that were available to EPA. And we kind of came up with this concept of a potential bioaccumulation model domain. And I'm going to try to see if I can get this. Oh, there we go. Okay. So at the core of the model is the bioaccumulation, food chain, and food web, which we've kind of looked at. Then there's a potential for the impact of uptake chemical bioaccumulation and toxic and effects. Now, some models will model the chemical fate, and others will sort of prescribe the chemical fate based on an external model or based on data. So there's a difference. Then you have the eutrophication piece, which we discussed was nutrients and organic matter impacts, some of the driving variable in terms of hydrodynamics, et cetera, and metals and sediment geochemistry. So when we looked at this potential domain against the AQUATOX domain, what we found was that it really has a very broad domain compared to, it probably had the most orange squares here filled in of any of the models that we looked at. But some of this points out some of the things that it does not do. It does not model the fate of metals. So there's issues of metal speciation, methylation for mercury, and metal sorbing directly onto organs. There are some models like the biotic ligand model that only do metals and metal speciation, and so their whole domain is this little corner down here. But that's not within the AQUATOX model. It doesn't explicitly, it does not explicitly model bacteria or pathogens, those microbial processes are implicit in the model and decomposition rates. And it does not model temperature, which is not included in this, so it doesn't model changes in temperature, but that's a driving variable that does have a very significant impact on model results. And it doesn't model fine-scale hydrodynamics. So there is a sort of simplified hydrodynamics component in there, which will define water volume and water velocity and the like. But it's the AQUATOX segments tend to be large, and so most hydrodynamic models have much finer grid cell. However, it is easily linked to existing hydrodynamic models, and that's been done many times, and I'll talk a little bit more about model linkage going forward. So I think in terms of the potential applications for AQUATOX, I think we've covered this a little bit, but nutrient applications, toxicant substances. So for nutrient applications, we are going to show a case study of a TMDL that was created in part using AQUATOX simulations as one important level of evidence. Toxic substances, for bioaccumulation, that's a little bit straightforward. We are also going to show a case study where it was used for a natural resources damage assessment. Some other things that it can be used for could be invasive species. Analysis has been used for that. There's a paper by Tainer where they look at climate change impacts on ecosystems using AQUATOX. And again, a multi-stressor analysis is an important strength due to the breadth of the model domain. So in terms of the regulatory endpoints model, I'm going to spend a lot of time talking about the AQUATOX output coming up, so I'm going to jump over this slide pretty quickly. But nutrient and toxicant concentrations in the water or in organisms, and chlorophyll A is an important one, and paraphytic chlorophyll A, which relates to the TMDL that we're going to talk about, are some of the ones to highlight there. In terms of how AQUATOX works, all of the state variables in the model, which include the biomass of all the animals and the plants, the nutrient concentrations, and the chemical concentrations within the animals and the plants, they are all solved in a simultaneous system of differential equations. The default is to use a variable step Runge-Kutta, which is kind of an efficient way to solve things, because when the model's running pretty smoothly, when there are no big disruptions, it runs with a larger time step. When there are disruptions in the system, it really cuts down the time step to get accurate. But it can also use a fixed time step as well. Usually it's on a daily reporting time step. You can use hourly as well, and within the model, there's a lot of options to change the averaging period to monthly or even annually, if that's the kind of output that you want to look at. And the model is open source. The code is all available. It was written, so it's showing a little bit of its roots in the 1980s in Pascal, object-oriented Pascal, but it is currently being ported to C#. And it's going pretty easily because the model really was written in an object-oriented manner, such that it's easy to port to another object, sort of a more purely object-oriented code base. And in terms of the processes simulated, I just want to say here that both biotic and chemical processes are modeled. And it really looks at direct and indirect effects, and it can look at the implications of the disruption of the food web, either because of nutrient or chemical perturbation or any other perturbation. That you can also externally add a perturbation to the food web and look at that. So the AQUATOX interface was developed started to be developed in the mid-1990s when everything was moving to a Windows platform. And so it has a pretty large, given that it has a pretty broad domain, it has a pretty large interface. So you have this, when you bring up the model, one of the simulations that kind of comes up with a main screen, and you've got a list of state variables here, too small to see here. Double-clicking on any of those state variables brings you into a loading screen, which allows you to add point source or non-point source loadings for nutrients. or other state variables. Now, some of these are linked to underlying databases, so they have sets of parameters that go with it. For example, plants, chemicals, and animals. And so you, you can go further into the state variable and look at the underlying parameters. And one of the things that in our design considerations for AQUATOX was to basically expose as many of the parameters as we could to maximize flexibility for the modeler, and also maintain transparency in terms of what the assumptions are, in terms of what's driving the model. Now, unfortunately, that also can just increases the amount of information that's available, which, as we all know, isn't always a good thing in the internet age. So it can cause confusion or feeling overwhelmed. But that's kind of the way that it works now. And we actually have some guidance available on the web for AQUATOX application and setup, and in terms of which parameters you should start with and which parameters you can leave the same, which are site-specific, and which are sort of the sensitive calibration parameters. So once you kind of learn, work with some of that guidance, it becomes a little bit less overwhelming. The majority of the parameters can just be left intact. Let's see. Okay, and you can also link through to the site. Obviously, you can do study setup and set the timing of the day, and there's sensitivity and uncertainty analysis. Another important piece of the model, a strength of the model, graphical interface, is the capability to graph the model outputs. It's a very flexible interface to do this, and it has the capability to import observed data, which is really nice. You can just run the model and see how it's fitting the data, run the model again, see how it's fitting the data. Within this capability, when you load a simulation, it's also going to save all of the graphs that you created for that simulation. When you save it, it's going to save all the graphs that you created for that simulation, unlimited number of graphs. So if you're calibrating to nutrients and organic matter at the same time, as well as chlorophyll A, for example, you can have all those graphs. And one of the things that I like to do is it gives you the capability to export all the graphs simultaneously to Word, so you can have a record sort of your evolving model calibration. So a lot of capabilities there. And then also within the interface, there's a sensitivity analysis which can help you understand which of the parameters have the biggest effect on model simulations. In this case, you select a dozen parameters, and then you change them by 20% on a one at a time basis. So that's what a nominal range sensitivity analysis is called. Is how that works, and you can look at using this tornado diagram concept. You can look at which parameters have the biggest effect on the output that you're looking at. In this case, periphyte and chlorophyll. But if you're not satisfied with the one at a time analysis, there's also an uncertainty analysis in which you can represent all of the point estimate parameters in the model as distributions, and then run an uncertainty analysis and get sort of a range of outputs based on uncertainties. That can be used either for sensitivity analysis or uncertainty analysis. So what has become clear is that this powerful graphical user interface is both a strength and a weakness of the model for a number of reasons. One reason is there's a bit of a learning curve. If you've been working with it for quite some time, you can become very efficient at changing model parameters and looking how that affects outputs, exporting things, importing things. If you want to import, dozens of time series simultaneously, there's a linkage to Excel to do that. So there's a lot of power to it, but there's a significant learning curve. Maybe more importantly, it reduces the interoperability of the model with other models, other modeling systems. A lot of those have ASCII input-output. Some people like to run their own auto-calibration tool or a sensitivity analysis tool that is really expecting a text input-output. So to remedy that, as of release 3.2, AQUATOX does have ASCII input-output where you can modify the model, you can run the model, you can examine the output. Everything comes out on an ASCII basis. So that, when we know some people that are actively working with that right now for their own sensitivity analysis or incorporation into linked modeling systems. The other piece that I want to discuss here is the recent work that we're in the process of in terms of adding AQUATOX into the EPA hydrologic microservices project, which breaks the model down into a number of individual web services, and basically taking all of the submodels within AQUATOX and cutting them out so that you can either run the full AQUATOX model via a web service, or you can have interoperability if you want to use a different water volume model, but you want to use the AQUATOX nutrient submodel. You should be able to, the design of the hydrologic microservice, because there's a number of other models that are also in the process of being brought into that environment, should allow you that kind of interoperability and picking and choosing what you want to use. As part of this, also, the code is being translated into the more modern C# programming language, and the web services are run on a .NET framework, and all the input of the model is being converted into a JSON input-output, which is actually the object-oriented nature of AQUATOX makes it pretty easy to export to JSON, because it's basically an AQUATOX segment object, and it has all these other objects in it, so it can be sort of a single command. Just save that AQUATOX simulation, it all gets written to JSON. So it's been a fairly smooth procedure so far. The other thing to note is that as part of this, to make sure everything's, the code translation is going correctly, we've been precisely replicating the results from AQUATOX 3.2 in that environment for each step. But the thing to note is that it is under development. These pieces that are pictured here are what are currently in the model, but they're being tested. And the plant and animal models are still under development. So kind of keeping in line with the expose all of the parameters design concept, which can sometimes be overwhelming and sometimes be powerful. The AQUATOX output has the potential to be overwhelming, so I want to just focus on a few of these that are most important. Obviously, the concentration of the state variables, the biomass, the toxicants in water, nutrient concentrations, those are important. One sort of value-added output that comes out of the model can be biological metrics such as percent EPT or trophic state indices, so you can look at the impact of a nutrient change on a trophic state index, for example. For bioaccumulation, obviously, the parts per billion of a toxicant within an organism is important. The other thing to point out here is that AQUATOX does balance the mass of chemicals, nitrogen, and phosphorus, and part of the way that we ensure that that's taking place across all these additions and losses and state variables simultaneously is through some mass tracking variables. And the other piece which I'm going to talk about next are state variable rates. And so, as I said, the way the AQUATOX structure works is that each one of these components of the model is a state variable, which means it is reflected by a differential equation. So in this case, we're looking at a differential equation for periphyton, bottom-dwelling plants. And so when photosynthesis occurs, the biomass goes up. When algal respiration occurs, the biomass goes down. When predation occurs, it goes down, et cetera. And so, again, in terms of at the risk of being overwhelming with output, all of these individual rates, these individual pieces, these kinetic rates for every state variable may be saved and graphed, and that sort of helps a user again, look under the hood and not have a model just be a black box. Okay, why, for example, are we getting these predictions? And this is kind of a messy graph for the first graph, output graph to show for the simulation, but this is showing a periphyton example where this dark red line with red circles is the periphyton biomass, and the other, which is in grams per meter squared dry, and these other pieces are the rates that are associated with that. And so you can see that when the periphyton mat builds up too high, and there must have been a low flow event here, but then you had a high flow event and you have sloughing. And so the question is, if you saw this peak and you said, well, why? why do we have this collapse? You can go into the rates and you can see that you have this this sloughing event occur. It has been predicted and is occurring. Whereas over here, the reason that things are going down is because of this olive green line, which reflects predation, and you can look at photosynthetic rates to see sort of what season it's doing well. So you can really look under the hood, you know, for better or for worse, but you can get a lot of information about what's going on. So I'm going to go through the primary AQUATOX submodels. The ones I'm going to look at are plants, animals, nutrients, and chemicals. So the plant types that are included within AQUATOX include free-floating plants in the water column, like diatoms and greens. Includes rooted and free-floating macrophytes. It includes cyanobacteria, formerly known as blue-green algae, which can be an important regulatory endpoint. And it also includes what we sort of started with was these periphyton mats that grow at the bottom of river systems. These are some periphyton photos from the lower Boise River, which we're going to be discussing. And actually, even within the periphyton community, you can have diatoms, blue-greens, and greens. So there's a lot of variety, even within a single one of these compartments. We already looked a little bit at the plant derivatives. They're slightly different for the free-floating plants and the bottom-dwelling plants. The free-floating plants will wash out of a system. The bottom-dwelling plants will stay in the system unless there's enough, unless the conditions, the shear stress is predicted to make those slough. And we have, as I said before, when you click on a plant state variable, you can get to its underlying parameters, and there are a number of these. These are now available also as SQLite databases as of release 3.2. So that is one product in itself of the AQUATOX model. Is these databases of parameters with associated references that can be accurate, and then you can all these reference. There's a list of all the references that are used on the EPA website, so you can go back and figure out what this means. You know, to find the precise reference. And as I said, there's we've starred some parameters here that are the most important. If you're calibrating or if you're trying to add another similar type of organism, you know, for example, the maximum photosynthetic rate, that can obviously be important because that's related directly to the doubling time. And one of the things with AQUATOX plants is that with ecological modeling is that it's hard to to use a black box approach for something like a plant because you're going to have species adapting to different temperatures, for example, and you're going to have species adapting to different nutrient levels as well. So there could be, you know, a diatom, and there could also be one that loves high nutrients and has a very high doubling speed and thrives under those circumstances. But in another site that's lower nutrient, you could have a diatom which has different characteristics. But we have, within the parameter set, which is over 60 records, we have a lot of variety to work with. So looking at periphyton. Periphyton is one of the unique components of the AQUATOX model. A lot of models include floating algae, or they include, but there's fewer of them that include periphyton. And so this model was put together utilizing an extensive data set from a series of experiments that manipulated nutrient levels, looked at different light levels and grazing pressure by predators, snails in this case. And so this is an example of two simulations against that. I believe there was 19 different simulations, and Dick felt like his parameter set successfully was calibrated against something 16 or 17 of those. In this case, we have one case where you have snails present, and so you really don't have periphyton building up at all. You have a very low level of periphyton throughout the whole time. When snails are removed, you have an increase of biomass. Then you have this sloughing event that occurs, and Dick actually talked to the people who are doing this experiment and said, did sometimes in your experimental facility, did you just have the whole mat lift up and move down? And they said, yes, that did actually happen. So he felt, I guess it would also be hard to describe how you have this massive decrease in biomass that's observed unless you have some sort of sloughing event or other kind of die-off. But that's kind of the basis of the periphyton model. And that brings us to one of our application cases that I want to talk about, which is the TMDL for Lower Boise River, Idaho. This was improved in 2015, and one important line of evidence was an AQUATOX analysis. And Idaho DEQ was the lead model in putting together this application, but we had an existing Lower Boise River model that they modified that we put together with EPA funding several years earlier. I just want to point out there's a very large watershed, and then within AQUATOX here we actually modeled 63 river miles of the Lower Boise River. And this is probably the most complex physical setup that I've ever done with AQUATOX, which was 13 linked segments. And while that's not completely unusual, the number of rivers that you have coming in, the number of agricultural withdrawals, wastewater treat point source and non-point source groundwater pieces was very important. And this river is highly impacted by agricultural withdrawals and returns. For example, the estimated natural flow here would be these red bars, but the actual flow due to natural agricultural withdrawals are these light blue bars. And then you often have the returns from the agriculture, which are very highly have very high nutrient concentrations. And so what we did within this AQUATOX simulation was we modeled the main stem as sort of as state variables, and we had all the inputs coming in exclusively. And when we had many dozens of different inputs from different data sets. This was a very data-rich environment, which was extremely helpful. Each little tributary had nutrient loadings that we could work with. But I was also extremely pleased that when we put all these into the main stem, that we did end up with really high-quality calibrations to nutrients. And so the Idaho DEQ took this model and they did some calibration to observed periphyton, and they actually simplified the model and did not include animals in it. So they modeled grazing pressure sort of as an external rate rather than including it. So, but here's an example. In a location above the city of Boise where you have pretty low nutrient concentrations, and you also have a low periphyton concentrations. And then you get down below the city of Boise after there's a lot of these nutrients have been added. The AQUATOX simulation is this orange bar. And the observed data that is temporally relevant are these black symbols, and the purple symbols are sort of the historic data sets that they have for periphyton. So you can see that the model is calibrating fairly well against this nutrient gradient, which gave Idaho DEQ confidence that it could be used to look at what happens if you reduce these nutrients. And of course, there were many different segments looked at. I'm just showing two of them. So Idaho DEQ was trying to answer the question, how can we meet the nuisance aquatic algae target of less than 150 milligrams per square meter? And they ran a number of different conditions. They looked at different best management BMPs for agriculture, and they looked at point source and non-point source loads. And they had a number of scenarios was sort of increasing stringency in terms of reducing the concentrations. And and this should say Idaho DEQ. My apologies to the DEQ. They found there was a point of diminishing returns in TP reduction across these scenarios, and there was the AQUATOX model was predicting that periphyton dropped quite a bit once you get to scenario 3, and then there was not a whole lot of additional reduction at that point. And they also noted that the periphyton was a complex community of algal groups, and it responds to these changes in a species-specific way. And so they had they simplified the model such that they took the animals out, but they added some more complexity in terms of periphyton. And I think that's one of the things that I haven't mentioned, is that you have a lot of control over what state variables you can include, whether you want to include a single algal group to represent all algae, or whether you want to break that out into 4 or 5, or even for a single piece like here, like periphyton, whether you need to consider the high nutrient and the low nutrient periphyton as part of your simulation. So you can make a very simple simulation to a very complex one. And in this case, they added the complexity where they needed it, and they took out the complexity where they did not need it. Another nuisance algae is the cyanobacteria, and the model has some very specific assumptions in place about where it's located in the water column, the impact of temperature, and the maximum photosynthetic rate. And we've done many calibrations of AQUATOX across blue-greens, and this is one case here, which is from a paper from Jim Carlton, where there was in Minnesota rivers, they modeled, looks like about a dozen different stream segments, and compared that to the observed data from those segments, looking at the impact of the blue-green index against the TP. And so the model is getting the general shape. It looks as though the model may be predicting blue-greens that are a little bit too high in the upper TP range, but that would give you some sense of observed versus modeled data. In that same paper, they looked at chlorophyll A results over time, and so this is an example from the Blue Earth River, which is a highly cultivated agricultural watershed. It has very high nutrient and sediment loads, has high chlorophyll A concentrations, which the model picked up definitely very well in the year 2000. Was a little definitely over predicting in the year 1999. If you look at that same parameter set in the Rum River, which is which is much less impacted by nutrients and turbidity, but it is also a very flashy river. So it's shallow and it has a high flow. So the model is the observed and the modeled data both drop down by, it looks like, about a factor of 3 or more. And on the basis of this, there is a technical note on the EPA website about how to about the parameter set that has been modeled over a nutrient gradient that can be used and has actually been brought into other sites. I think actually it was used as to start the Lower Boise River, Idaho, and also was validated using the Cahaba River, Alabama simulation. So there is a real starting point for calibrating chlorophyll A across a gradient. The other thing to point out is that there is an internal nutrients option within the model, also called luxury uptake, where algae will take up nutrients that are in a period where it does not actually need those nutrients for growth, and save those for periods when there's lower nutrient And so this is in the model following the approach of Qual2K and WASP-7. And so this increases the number of state variables and the complexity to some extent because each nutrient in plants have to be modeled separately. I think that brings us to our first break for questions.
Thank you, Jonathan. Before we start questions, I just want to note that I understand there were some technical issues at the beginning of this webinar, and that caused some people to join late. And so I wanted to mention again that this webinar is being recorded, and a link to the archived file will be available on the Modeling Workgroup website. The URL for that is shown at the bottom right of your screen. That'll be available in a few weeks. So Jonathan, first of all, I have two questions for you. The first question, you said that temperature is a limitation, but AQUATOX can assess for climate change. Can you talk a little bit more about that?
Yeah, so it's a limitation in that the model is not going to say, okay, taking into account the amount of sun that is hitting the river body and what the water that's coming in and the retention time, it's not going to calculate the temperature of the water. But when it's been used for climate change, there have been other models or other assumptions in terms of how much it's a driving variable. So you can modify the temperature of the water based on either assumption or observed data or additional models, and you can get that answer from climate change. So hopefully that clarifies that question.
Thank you. Second question. Oh,
yeah, go ahead.
Has AQUATOX been used for modeling toxic microsystems in aquatic systems?
It has not yet. We've kind of just been looking at total blue-greens rather than sort of breaking out those blue-greens which cause these toxic effects and those which don't. And I think we've looked at it, and I think that there's still it's complicated in terms of what precipitates the algae to create the toxins and create those toxic events. And Brenda, I think you might have looked into this a little bit more than I have, but maybe not.
Yeah, thanks, Jonathan. Yeah, I'm going to ask, this is another one related to that. Does the model account for cyanobacteria that can fix nitrogen versus those that cannot?
Yes, I believe that it can. It has nitrogen fixation is something that is in the model for some organisms under some circumstances. So I would say yes.
Thanks. And then how can we use AQUATOX for ERAs, which I assume is ecological risk assessment?
Yeah, that's an interesting question. I mean, I think the answer would be to sort of utilize that core of the model. What I've seen in terms of ecological risk assessments is that what they need from a model is the tissue residue, so the concentration within tissues, and then the ERA runs from there and calculates hazard quotients and the like. And AQUATOX can give you that. I mean, I think AQUATOX can also give you the effects. So it can, if you have toxicity data, it can calculate some impacts. But that's beyond what I've seen most ERAs use. I think usually they're just looking for the concentration in tissue. So that would be what I would think would be most ecological risk assessors, how they would want to use the model.
Great, thanks. And then next question, how can a user decide whether a calibration is successful, and does AQUATOX provide tools for this decision?
Well, I think that is an interesting question, and it's obviously a line of there are a lot of lines of evidence simultaneously to look at with respect to that. I mean, it kind of depends on what you're calibrating against, how strong your observed data are, We actually have we have a 3-day course where we talk about the application of AQUATOX, and there are some discussions of the different lines of evidence. I think one of the most important tools is comparison of observed data versus model results. So that's sort of your first level, and obviously all the output can be exported into other statistical tools to calculate absolute error, mean absolute error, or things along the lines of that. And then I think sometimes, I guess, looking at uncertainty analysis can be helpful. So I think there are tools there, but I do think, there's no, because there's so many different types of calibrations, whether you're calibrating to nutrients or calibrating to biomass or calibrating to chemicals, that to have like one tool that says you're there or you're not there would be overly simplistic. So unfortunately, that's where the sort of art of applying a model has to come into play to a certain extent, art and science.
Good, thank you. Next question. When discussing the Boise TMDL development, you showed that light availability was an active variable. You may have just answered this, but does modifying this variable influence the water temperature, thinking towards thermal credit trading like Metford, Oregon?
Right, and so the answer is that temperature is strictly a driving variable at this time, so you would need a different model to look at that. So the model does spend a lot of time working on light climate, and it looks at the impact of organic matter, inorganic matter, algal blooms, and tree canopy has on the light, and it does different calculations for light climate, the top of the water and the bottom of the water. So and the Secchi depth of the water. Those are all sort of critical, but at this time, none of those have an impact on temperature because that's just something that comes in from outside the model.
Okay, thanks. Next question. What is the advantage of using AQUATOX over EcoSAR, and in parentheses, EpiSuite?
I don't know the answer to that.
Okay, thanks. There's one more being typed. I'll wait just a minute. Okay, and then last question I'll ask during this session. Are there other endpoints other than chlorophyll A, like cell counts, biovolumes, microsystems?
Well, thank you. That is a good question because it brings me to a point that I haven't made that is an important point about this model. In that AQUATOX is really it is a biomass model. So it calculates all of for animals and for plants. It calculates the total mass of the animal, total mass of the plant, and this is how it balances mass and balances the food web. So there is a biomass component. That is another output. And there is, biomass of the different types of plants and so percent blue-green like that. But because it does not a population model, so it's not going to give you the number of fish in that case or cell counts in this case, it's not going to directly give you bio volumes or microsystems.
Okay, thanks very much. So
Sure.
Jonathan, why don't you go ahead and continue with your presentation. Thank you.
Sounds good. Okay, I'll plow along into animals. And I think we kind of looked at this a little bit before, but there's quite a variety of invertebrate types, and then fish types as well, broken up into bottom fish, forage fish, and game fish. And for the fish, we do calculations of their biomass using the predictions of bioenergetics models, originally Wisconsin Bioenergetic, now Fish Bioenergetics 4, to predict things like growth rates and respiration rates and food consumption rates. The other thing to point out is that we can model, I said it is a biomass model, but we can break out and we can model size classes. So fish can be modeled as juvenile and adult, for example, which can have very different characteristics for bioaccumulation and feeding preferences and the like. And then there's also, it tracks as they get promoted from one class to the other. It can also be modeled on an age class basis, which, you're starting to add a lot of complexity to the model, so you don't, I mean, it's worth doing in many cases, but you have to recognize that if you're modeling 12 age classes, now you have 12 times the amount of output that you had previously for the fish. And if you have the data to support that, then that's good, but sometimes that is not the case. Very much like the plants, the biomasses are calculated dynamically. There are a number of allometric equations that are used along with the bioenergetics parameters to calculate different kinetic rates associated with these derivatives. So here's an example of the animal derivative, where you have you'd have loading, where in some cases you might actually have an external load or upstream load that comes in to play when the animal consumes, it gains biomass, and it defecates, it loses biomass, etc. Some animals can undergo migration. Some animals maintain their place in the water column. Others wash out of the system. And these are all obviously mortality, predation through the food web, etc. And just like the plant, there is an extensive animal database that's available, and with references and some parameters that are more important than others. Optimal temperature being one of the ones that drives the calibration in many cases. Like all food web models, there has to be some specification of feeding preferences within the food web. Different than many bioaccumulation models, because biomass of these different compartments varies, there is an opportunistic feeding component. So if you have if you have a game fish that has three prey sources, but then one of them has had an impact for some reason, whether it's a chemical impact or other type of impact, and the biomass crashes, it's going to switch its feeding to the other species to some degree. So it tries to maintain ecological realism and sort of a mechanistic component in that manner. That was a very brief overview of animals. I talked a little bit about nutrients, and I put this last, even though it's sort of, in some ways, it's the simplest, but because it is very much impacted by the animals, the plants, and the organic matter, which in AQUATOX we call detritus. So this is a schematic of the nutrient cycle in AQUATOX for nitrogen. It's almost exactly the same for phosphorus. You have assimilation by plants, then you have return to the water column through excretion and respiration. You have decomposition of organic matter can result in a significant difference in the nutrients and the like. And then there are interactions when an animal eats a plant, the nutrients are transferred and the like. And again, as I said, the mass balance of nutrients are maintained through all of this, and there are specific checks in the model for that. Another piece that I wanted to discuss, because it's become very important in terms of our application of the model, is the sediment diagenesis model. So this does add some quite a bit of complexity, because generally in the organic matter, I don't spend a lot of time talking about organic matter in this, but in the organic matter and the sediment bed is reflected by a much smaller number of state variables, reflecting sort of slowly decomposing and more quickly decomposing organic matter, and then also dissolved organic matter in pore waters, potentially. But in this case, you've got, you actually are modeling a two-layer sediment bed with a very thin aerobic layer and a larger anaerobic layer under that. And this really provides a lot more information in terms of and you have 3 different reactivity classes for 3 different state variables. So the number of state variables multiplies quite a bit. But you have the mineralization of methane, and then you have oxidation causing sediment oxygen demand, and you also have the nitrification of ammonia explicitly modeled, and that causing sediment oxygen demand. And so what we found when we were modeling some systems that are really highly impacted by nutrients, for example, when we were modeling the 10-killer reservoir in Arkansas, we were really having trouble with our sediment oxygen demand and our oxygen calibrating the model to oxygen levels in the hypolimnium. That was a vertically stratified reservoir, and we modeled it with I think 8 different segments. But we were having trouble getting the model to fit, and we essentially turned on the sediment diagenesis model, added all these state variables. There's really only, again, when you add this to the model, there are dozens of parameters that are now exposed, but there are really only a couple that are considered to be site-specific by DeToro. And we didn't actually have to do any calibration, and all of a sudden our oxygen concentration just snapped right into the data. It was really, so it's a very impressive model. And so it should be considered. The other really important thing about this model, sediment diagenesis model, is that it takes into account the fact that when you have oxygen in the water column, that this diffusion of phosphate into the water is limited by because there's essentially a protective layer in the aerobic layer when phosphorus sorbs to the ferrous iron, and when the system becomes anoxic, so when your oxygen in the water column goes down to 0, that disappears, and all of a sudden you have a major flux of phosphate. So the diagenesis phosphorus flux is much more accurate as well. So as you can tell, when it's needed, I think it's a really good model. Moving on to discuss organic chemicals, which was really kind of the what Dick had in mind, and when he created AQUATOX and combined all these models together. So you can model a lot of chemicals simultaneously, up to 20. There are a number of in terms of chemical state, in terms of calculating what's going to happen to the chemical, there are sort of a number of fairly standard processes that are part of the differential equation. I will note that you can model biotransformation as well, the daughter products, if you're specifically including those daughter products in the simulation. When you get into the chemical derivatives, things can get very complicated because there's so much interaction between and sorption to every different compartment, whether it's the organic matter, sorption and desorption, whether it's direct gill uptake into the organisms, etc. And then each compartment, whether it be a plant or an animal, has an associated chemical with it. So those 2 variables are calculated independently. And again, as will be no surprise, you can plot these rates, and this is a sort of a simple case where there was a chemical called HCB in a tank, and you can see that the concentration of the chemical drops. This blue line drops quite a bit, and the reason is primarily volatilization and sorption into the plant, because this was modeled with a macrophyte in that tank as well. That's just an example of sort of how you can do a very simple model to test the constructs of the model as well. You can see what the concentration is within the plant, and you can see the concentration in the water, and look at all those things simultaneously. And the bioaccumulation model within AQUATOX, you start with this calculation of the toxicant in water. And if you're looking at a game fish, for example, there are a couple of primary routes of uptake. There's the uptake through the gill, which is calculated. Then you have partitioning to organic matter and other food sources. So you have dietary uptake, which takes into account the fish bioenergetics and consumption rates, chemical assimilation efficiency, and there's growth dilution. So there's a number of factors that come into that model. But it's very, all of these pieces you're going to find in really any bioaccumulation model, sort of a standard bioaccumulation model construct. I do want to spend a moment and talk about the perfluorinated surfactants model that is in AQUATOX. This has been in AQUATOX for a while. It was added in 2005, and it was based on the best data at the time were from some publications from Martin and coworkers in 2003. And so there are some simplifications. The kinetics, the sorption of PFAs to animals and organic matter in plants is primarily modeled using an empirical approach, and the uptake and depuration is a function of the chemical chain length and the PFA type. And so here are some of the data that Martin had showing that as your perfluoroalkyl chain length increases, the K1, which is your uptake rate, increases quite a bit as well. So you have a higher uptake for a higher chain length. And in the next slide, we see that as your perfluoroalkyl chain length increases, this is a K2, which is a loss rate, a depuration rate. And so that decreases as your chain length. So the upshot is those PFAs, which have a higher chain length, are much more highly bioaccumulative. And so there are a number of data requirements for the PFA model, some bioconcentration factors. The chain length, as we talked about, is a very sensitive parameter. If you want to look at the impacts, there's some toxicity data, but I will note that there are parameters in the database that we've gathered and worked with for PFAS and PFOA. And we had some testing of this. Brenda Rashley in 2012 put the model through some testing. So this is based on the Conasauga River in Georgia, and started by estimating PFAS concentrations in the river based on a log regression with discharge. So had a pretty strong relationship between PFAS concentrations in water and discharge, and then applied that to some discharge throughout the period of the simulation. If you take that and then you add the food web to it, you can see that these are some concentrations of concentrations in organisms in the food web in parts per billion. So if you look at this red line for bass, it during one of it gains some PFAS and then declines, goes back up again, pretty high, and then it's in a process of depuration when it goes through the observed data, and it goes through the observed data fairly well. The catfish has a very similar curve. It kind of exceeds the observed data. Potentially could be a timing thing because it's in a sort of a state of pretty rapid decline there. And it looks like there's a good fit for oligochaete and maybe an over-prediction on the mussels. So this is I don't recall or know how much calibration took place there, but it shows that the model at least has the capability to calculate the bioaccumulation, certainly in the right order of magnitude of what's going on in the system. Looking forward with this model, there have been a lot of recent studies and publications on the mechanisms that govern the bioaccumulation of PFAs. And my hope and my plan for the model is that we can continue to test and refine the model using those data. And one of the pieces that we may need to incorporate mechanistically is interactions with proteins, which is something that is already in the AQUATOX model. The proteins are. Phospholipid is not, but I do see that there are data available in terms of phospholipid, and sometimes there are regressions available between lipid and protein and phospholipid concentration. So that may be a potential step forward to make it a little more, to refine it with the most updated data. So in terms of, then we have that bioaccumulation, then you have the impact of the chemicals. And so the model can look at both lethal and sublethal effects. So LC50s and EC50s are in there. The sublethal effects would be growth and reproduction impacts. One of the things about the model is that it uses what's called the critical body residue approach. And so what this approach does is it calculates an internal LC50, a concentration where LC50 means a concentration where 50% of your organisms observed had died. So it calculates the internal LC50 based on two lines of evidence, the external LC50, which is measured using laboratory studies, and the bioconcentration factor, which looks at the relationship between an internal concentration and the external concentration of an organism. So if you combine those together, you can get LC50 internal. And the reason that that's important is that it takes into account the bioaccumulation in terms of what toxicity is going to be predicted. Because most toxicity studies are just looking at, you have the they don't take into account this feeding mechanism and the bioaccumulation in terms of the impact. It's just you have a certain amount of chemical in the system, and then you have a certain amount of the organism die. But on the other hand, sometimes, for whatever reason, an external toxicity model is what you want to work with, and so it has flexibility to do that as well. Another piece about toxicity is that if you're modeling a food web with, say, half a dozen animals, you may only have toxicity data for 2 or 3 of those, if you're lucky. And so one way to fill in those gaps is to use this EPA product called ICE, which is interspecies toxicity correlation. And it basically if you have an observation from one organism, but you have and then you have another organism in your study, it can make an estimate of what the toxicity will be for that other organism based on all the chemicals that were tested for both of those organisms, and it will give you a sense of the uncertainty around that. It will give you the confidence interval in that prediction. So that's just a useful tool to help you fill in those parameters. Last thing I want to go through, well, it's close to the last thing, but I want to talk a little bit about the recent application of the model to a lot more saline environments. So AQUATOX 3.2 includes what we call the nearshore marine environment version. This did go through a successful peer review in 2015 and is published in the open literature, which in some studies that I'll show in just a moment. For example, we added a very specific oyster model, which has size classes moving from the veliger up to settling down as spat, maturing to sac, and then spawning back to veliger again. So it's a size class model that has gone through the peer review and quite a bit of testing. And the reason for the creation of some of these organisms that we've added and these constructs that we've added to AQUATOX, we already had an estuarine version, so we were already dealing with salinity, so it wasn't a major change to the model. Was that the model was recently used to look for natural resources damage assessment to model the impacts of Deepwater Horizon for Mississippi and Alabama. And there's a couple of papers that came out of that. The first one is Don Blanchard and co-workers where they did a baseline estimate for a number of different habitats in Mississippi and Alabama, a baseline productivity estimate. And appeared in ecological modeling in 2017. There was quite a team of scientists that we had working on this from DEQ, and University of Southern Mississippi, and consultants, and some real experts like Chet Rakazinski in particular, helping us make sure that we had realism in terms of the food web and the ecological interactions that we had in place there. There were a lot of, it was a fairly complex model. There were a lot of biotic compartments that were included there. Of course, this also includes all of the plants and the invertebrates as well. And some of these existed in the AQUATOX library, and some of them were added. So there's a nice marine database you can draw from. I spent a lot of time looking at the food web setup, outputting trophic levels that are predicted from the model and comparing those to those that were derived with stable isotopes. And the result of that was some baseline productivities in grams per meter squared for different habitat types, whether it be an exposed beach, a soft bottom. What we found was that the oyster reefs had the highest overall productivity in both Mississippi and Alabama. And some of these were compared against existing estimates of productivity, overall productivity for the different habitat types. So starting with that baseline productivity estimate, we could then look at the establishment of the nearshore marine injuries as part of the natural resource damage assessment. Same team was involved. And so the way that this put together was that we start with the baseline productivity model, which we just discussed, how that was put together. We added, there was some good information about TPAH exposure, and this was actually modeled with a number of different, we modeled it with 7 different compartments, breaking it up by KOW, and that also helped us to apply different toxicity data to it. So there's sediment data, water column data. Then we had a bioaccumulation piece, which very significantly took into place the metabolism of PAHs, which is an important component of bioaccumulation for PAHs. And there were, through the overall federal natural resources damage assessment, there was a tremendous amount of toxicity data that were available to work with. So if you combine all those elements together, the other thing I will note is that we did spend some time, to the best of our ability, working with bioaccumulation verification. One of the issues that we had was during the period of highest oiling, there was very little biotic sampling that took place. I mean, that was maybe not the first thing in everybody's mind as they were scrambling about. But in terms of both of these data sets, we have data from before and after, but not the highest peak of no data from when the sampling was occurring. But so we did some verified with the data, the best available data that we had, to make sure that we were getting the right degree of uptake. And as I said, because of metabolism, some of these organisms depurate the pHs pretty rapidly after their exposure. And ultimately, we resulted in an injury quantification by habitat type. And we found that the marshes and the unvegetated marshes had, not the unvegetated marshes, but the unvegetated nearshore marsh habitats had the highest injuries. But one of the interesting things was that because this is over a productivity loss over 3 years, because there was pretty rapid recovery of these habitats, and also because of the metabolism, and because these areas we were looking at, AQUATOX was being used to try to understand the impacts, not in one of the highest impacted areas in Louisiana, but in one of the areas where there was observed oiling. There was measured oil both in the sediment and in the water column. But there was a question, whether there were some that were advocating that Mississippi and Alabama had no injury at all. And I think based on our analysis, trying to take all these pieces together and be as conservative as possible, there was a non-zero productivity loss there. But also, under 4% is not the biggest perturbation either. And we could break it down in a lot of different ways and look at the quantification by species. The brown shrimp had some of the highest losses, and that was due to the toxicity data, the direct impacts of brown shrimp and the copepod. Anchovy also had some of the higher toxicity, the lower LC50s, so the higher impacts. And so some of these other are indirect. Another interesting observation was that the oysters, they did not show much. They had very high LC50s, and so we didn't show a lot of impact to oysters. And along with those publications, if you really wanted to dig in deeper, there are reports in the Natural Resources Damage Assessment. So my goal with this was to kind of, as I said, it's a broad model. There are a lot of topics that could be covered. And I wanted to give an overview of some of the things that AQUATOX could do, some of the science that's behind it, how we are using it both as sort of an integrated single unit model, but also trying to break out into submodels with the hydrologic microservices component, for example, or people borrowing pieces of code because it's open source. The movement of the code into C#, all those pieces together, as well as show some examples of how the model could be used in either format. I think that's my last slide. Oh, there is, just in terms of the case study that I was just looking at, I did want to note that the peer review, the EPA peer review was of the nearshore marine, the state variables, the biotic variables, and the model. So that was included in the peer review, not the application and the toxic effect piece. And just make the note that this case study presentation and model application does not represent opinions or endorsement by EPA. And that was my last slide. And I'll take any questions.
Okay, thank you, Jonathan, for that great presentation. As the questions start to come in, please note that when the webinar is over, an evaluation survey will be brought up onto your screens. We encourage you to answer this evaluation and give us feedback, as we intend to listen to the feedback and improve our webinars whenever we can. So we'll be moving into questions. So first question, is, can an LC20 or LCX be calculated instead of an LC50?
Right. Well, the model isn't calculating the LC50s as much as it uses those as an important input, a line of evidence to calculate the impacts, and it does on the basis of the curve. The LC50 can be input, but also the curve that would the slope of the curve can be input that would allow the impacts at a lower concentration, to potentially kill, say, 20% of the organisms. So having those data, the LC20, the LC50, the LC80, if you can have all those data, you can do a better job fitting your curve, and then you can have a better result out of the model. But it won't actually calculate those.
Thanks. Next question, is bioturbation accounted for in the sediment diagenesis model?
Yes, bioturbation is accounted for in the sediment diagenesis model, and it's actually interesting because DeToro has sort of a, that's one of the places where we move a little bit away from DeToro's model, because in his model he's not explicitly modeling the invertebrates, and so in the AQUATOX model there's the option to actually have bioturbation be a function of the biomass of the benthic invertebrates. Or you can use more of like DeToro's constant. I think he has a simpler relationship, but the linkage is a little bit. I think he just has a relationship to oxygen, and with AQUATOX you can actually use the biomass of the benthic invertebrates to calculate bioturbation. But in any case, it is accounted for.
Great. Thank you. Next question. Does AQUATOX generate outputs such as CSV or text files compatible with statistical software such as R?
Yes, it does. It can output the data in a number of different formats, Excel as well, but also CSV and text. Yeah, so we as well as, yeah, it has a lot of flexibility in terms of input and output.
Thanks. Next question. Can you briefly compare AQUATOX to other models such as PC lakes or WASP?
I don't always have the widest, as is clear, you know, I have a lot of experience with sort of some strict bioaccumulation models. WASP, I have worked with, but it was quite a number of years ago, and I know that they've added a lot of capabilities in terms of modeling, different algal compartments and the like. I don't know what the organic chemical bioaccumulation looks like in WASP or whether that is there, and I've never worked with PC lakes. So I probably, if you want to email me, I could probably do that at some point, but I can't off the top of my head. I could compare it to an older version of WASP, but I don't think that's particularly useful.
Thanks, Jonathan. I would say briefly that WASP is more spatially explicit but doesn't include the higher trophic levels like AQUATOX does, and that's something I can follow up on as well afterwards.
Yeah, thank you. I think that's a good. That's a really the spatial explicit is a really good point, and that's part of in terms of the way that AQUATOX works. It's very explicit in terms of food web. But because you're modeling an entire food web together, you're often looking at segments that are 1 kilometer by 1 kilometer or something, or a 1 kilometer river segment sort of at the smallest. Because I mean, it can be used to model terrariums as well, but if you're working out in ecology, usually your bioaccumulation model, your food web model, is going to be a larger domain.
Good, thanks. Next question, is there any library which the ready files for fish could be downloaded from, or any comprehensive sources which the fish data could be gathered from?
Yeah, I think if you, I think probably the best, I mean, there's a couple of things. You could just email me and I could email you the databases. We can the whole fish library. I can I can export all those parameters to Excel very easily. I believe it's an option right within the interface to just export the whole thing to Excel. It's in SQLite. If you download the AQUATOX 3.2, you can get them in that format. And then, as I said before, once you have them, there's some abbreviations in terms of what the references are, and there is a reference on the EPA website which kind of clarifies exactly what references are being talked about. But they are available. It's not like a product that stands alone, so it's not like a single website like let's download all the fish data, but it's pretty easily extracted, and I can help you if you need any help with that. Great.
Thank you. Next question, do the databases support use in tropical environments?
That's a good question. I don't think that there are a lot of tropical species in there. So I would say sort of not off the shelf that I'm aware of. Yeah. I think we could give some guidance in terms of how to create some of those tropical species. What parameters you might want to work with, how to start with surrogate species and create some of those tropical species. But yeah, I think the model has been well, I should put up one of Dick's favorite slides, which shows all the locations in the world where the model's been applied. It has been applied extensively in Europe, extensively in China, and throughout the United States. But I don't know of tropical applications off the top of my head.
Okay, thank you. So Jonathan, that's the last question, so we're going to move to the conclusion of the webinar. So should you have any additional questions for Jonathan, or if your question today was not answered, please feel free to contact him after the webinar. His contact information is included on this slide as well as mine. And you can also download a certificate of attendance at the link which is shown at the bottom right of your screen. Click on the certificate name and choose Download Files to download the PDF document, which you can personalize with the names of everyone watching from your location. More importantly, you can find information on future webinars as well as a recording of today's presentation on EPA's Water Quality Modeling Workgroup webpage. The link to that page is also shown at the bottom right of your screen. Today's presentation will be archived and available soon on this same website. So please visit EPA's Water Quality Modeling Workgroup webpage for details about the next webinar. And at this time, I'd like to conclude today's webinar. Thank you again to our amazing speaker, Jonathan Clow, and thank you to all of you for joining us. This ends our webinar for today. Thank you again for joining.